we get to it, you'll undertake a series of challenges to prepare yourself. Let's get started with your investigation.
This simulation uses voiceovers. If you don't have audio, you can access the audio settings by clicking the gear icon on the top left of your lab pad. Then select Audio and switch the...
Using the lab pad is perfect. Then let's get... Find the lockers and open one of them. Raise your lab pad for more information. Find a lab coat inside the lockers and pick... That seems a good fit. You look ready to make a start. Let's get to it. Think back to the last time you got injured. Was it a cut, a graze, maybe a burn from your new laser thrower? Humans are always damaging themselves. Luckily for you, your immune system is always there, monitoring and responding to health threats. Today, we've been tasked with part of a big global health surveillance project. Before we get to it, I need to make sure your immunological skills are up to the challenge. Infectious diseases don't just impact the people who contract them, they affect the communities they are part of. Health surveillance is the process of monitoring the occurrence of different healthcare-related events in different communities, things like vaccination, disease contraction, or even how often people go to the gym. By understanding the health landscape on a community level, it is possible to track how attitudes and illnesses spread from individual to community to country and even internationally. When the term global health surveillance is used, it means we're considering the collected data on a whole world level. The immune system is made of a variety of tissue and organs that give rise to a diverse cast of wandering cells. All are specialized in their own way. Some are detectors, some are capturers, and some are just natural-born killers. Really, they're actually called natural killer cells. Most of the time, the detector, capture, and killer cells do a great job of preventing pathogens from gaining access to our bodies. When those defenses fail, memory cells can keep molecular mugshots of the invading pathogens so we are prepared if they make a return visit. Your trials today start on a topic at the very roots of immunology. Remember those memory cells I mentioned when you first came in? Using a vaccine is like giving those cells a ready-made mugshot of a particularly dangerous pathogen. It primes the cells for response without the need for an infection. We'll learn more about this later, but for now, let's see how the vaccine landscape has changed over time. On the screen, I've collated global health data from a particularly interesting time in the Earth's history. The color a country is shown in indicates the percentage of children under 1 year old that had received a vaccine for a preventable disease. Lighter colored areas have the lowest uptake, and darker areas have better vaccine coverage. You can browse the data by choosing different years at the top of the screen. Browse all the maps shown on the data screen. Raise your lab pad when you've found a pattern. Are you finished exploring the map data? Did you notice how the world's vaccine coverage changed? It's amazing to see human progress in action. Cutting-edge immunological research and global medical access initiatives broadly improved vaccination coverage across the planet, saving many lives. Based on your observations of the map data, which of the following statements is true of the data displayed? You can lower your lab pad and check out the maps again if you wish to. Option A. All countries experienced a consistent increase in coverage between 1980 and 2015. Option B. Vaccine coverage improvement was inconsistent across the planet. Option C. There was a sudden drop in European vaccine coverage in 1995. Option D. Vaccination coverage was more unstable. Awesome observation. In general, people across the planet have benefited from better access to vaccines. Despite technological and humanitarian advances, factors like war, famine, and even personal beliefs can have a big impact on global coverage consistency. How do you think vaccination rate will affect the incidence of disease? Option A, decrease rate. Option B, no effect. Option C, it depends on the location. Okay, well, let's see if your thinking changes as you progress through today's challenges. I think you're ready to start your immunological investigations. I'll be here to check you've got the skills to have your access upgraded, meaning you can move to the next task. Remember, You can check your LabPad's theory pages if you need a helping hand. Today's final challenge will take skill, knowledge, and a critical mind. Complete each challenge to gain experience and immunological insight. Hopefully, this will help you in the final mission. Okay, I've just unlocked your security card ready for use. You can now use it with the card readers near the door to move between labs. Pick up the access card and swipe it on the card reader to start your first challenge. Place your access card on the far table. Excellent. The access card is your key to these challenges. When you successfully complete a challenge, I'll upgrade your security clearance so you can move to the next room. Make sure Before we deep dive into the immune cells and their interactions, I first want to make sure you're familiar with the organs of the lymphatic system. You'll need this info to complete future challenges, so make sure you are thorough in your explorations. As you can see, various organs have been displayed for you in our holographic friend here. Some are definitively part of the lymphatic system. However, others are simply closely allied. Before we go exploring organs, it might be helpful to fill any gaps in your knowledge. The body's primary defenses are the basic physical and chemical barriers that serve to keep pathogens out the body. Physical barriers are things like your skin. It keeps your insides in and the outside out. It can also include mucous membranes, like in your nose and mouth. Chemical barriers are often pathogen-destroying or moving liquids like tears, stomach acid, and earwax. Did you know your earwax has mild antiseptic properties? Option A. That makes sense. So what would the second line be? Option B. Where do immune cells fit into all this? Option C. I already know about the other lines of defense. Let me explore the organs. The second line of defense is our non-specific immune responses that are triggered by chemical molecules. A chemical signal of chemokine molecules might trigger an inflammatory response recruiting pathogen-eating cells to clear an infection. We'd also see swelling and feel pain. Signals with interferon molecules can warn other body cells of a viral attack, causing them to boost their defenses. Activation of a signaling system called complement can lead to opsonization, where pathogens are coded in signals and antibodies so specialized immune cells can destroy them. Option A. If that's the second line, what's the first? Option B. Is there a third level of defense? Specialized memory and specific response cells form the final level of defense against pathogens. B and T cells might be the most well-known of these. B cells have surface antibody receptors that can bind specifically to distinct molecular motifs called epitopes. An epitope is like a pathogen's molecular mugshot. Binding triggers a big immune cascade. T cells are slightly different. They recognize and kill any infected or malignant body cells. Option A. If that's the third line, what's the first? Option B. I'm confused about what the second line is if this is third. Option C. Immune cells are hardcore. I'm pretty confident with defenses now. Let me explore the organs. Great to hear you're feeling confident. Explore the different organs of the immune system. You've identified the heart, but what we're interested in here is actually the subclavian vein that runs underneath your collarbones. This important vein is where all the body's lymphatic fluid drains into the circulatory system. The lymph vessels form the circulatory network that lymphatic fluid moves around the body in. One-way valves in the vessels keep the fluid moving in one direction, squeezed around. I'm sure you're familiar with the two spongy masses at the back of your own throat, but believe it or not, you actually have a lot more. The two where you've seen them are called palatine tonsils. There's one above your soft palate called the pharyngeal tonsil or adenoid, and another pair in the base of your tongue called lingual tonsils. All of them serve to monitor microbial load at the busiest entrances to your body, the mouth and nose. Large intestine is home to lymphoid tissue residents like macrophages, eosinophils, and mast cells. They can phagocytize pathogens and release toxic and inflammatory mediators. Lymphoid tissues placed on the walls of large intestines manage the In the intestinal wall, there are specialized bundles of lymphatic tissue called Peyer's patches. They're responsible for monitoring the microbial load in the ileum, last portion of the small intestine. Like the nodes in your armpit and throat, these are specialized peripheral immune organs where lymphatic fluid is filtered and some immune cells become activated. Every immune cell in your body starts out as a stem cell from your bone marrow. Though we're looking at the femur's marrow, marrow can be found in a number of other bones, including the pelvis and sternum. That's the thymus gland. This primary lymphatic organ is relatively large in children, but then it shrinks as you age. That's not surprising, as the thymus is the site of T-cell maturation. As your immune system finishes developing, you no longer need quite so large a maturation organ. The spleen is a big immune cell reservoir that has important roles for red blood cells too. Among other functions, it nurtures a store of immature monocyte cells that can become dendritic cells and phagocytes. Sometimes when you're feeling run down, you can feel a swelling in your armpit. This is down to your lymph nodes enlarging as more immune cells are produced and matured within them. Sometimes you can also feel the nodes just under your ears and... Okay, let's see how well you know this system. What lymphatic component? Awesome! Fun fact. Peyer's patches are surrounded by specialized microfold cells that serve... What is the main role of the thymus gland? You can click the View Theory button to get some guidance with the question. Option A. It's a secondary lymphatic organ associated with the heart where immune cells mature. Option B. It's a primary organ of the lymphatic system where B cells mature. Option C. That's right. We'll meet the thymus and the T cells more closely in a later mission. What would happen if an otherwise healthy person had to have their spleen removed after an accident? You can click the View Theory button to get some guidance. The spleen is an important blood cell reservoir. It can release up to a cup of blood to replace red cells lost during intense bleeding. Up to a quarter of a It looks like you know your stuff. Nice work with the quiz. Review any immune organs you feel unsure about or raise your lab pad to continue. Are there any organs you'd like to look at again? Okay then. Congratulations on completing your first mission. I've upgraded your Pick up your access card and swipe it to take on the next challenge. Place the access card on the far table. In the next two challenges, you will come face to face with the cells of the immune systems, some of which were mentioned in your previous mission. We're going to figure out a few responses and roles of different. There are many ways a pathogen can create illness, so I've kept things simple in these challenges. In the first one, you'll only meet the cell. In the second challenge, things step up in difficulty. You will have to negotiate cells of the adaptive immune response as well as the innate. If you'd like to take a moment to learn more about the innate immune system before heading into the challenge, now is your chance. You can do this by clicking the View Theory button. I've also summarized the cells you will meet. Head over to the wall screen to view the cell summary, then tag the infection portal to take the challenge. How are you going to access the body? Option A, by inhalation. I'm going to enter via the nose. Yuck! What's that smell? You're definitely in the mouth. On the inner surface of the cheek, to be precise, and in the company of millions of bacteria and parasites. The immune presence is strong. You'll need to be careful. What do you do next? The saliva is awash with antibodies and digestive enzymes. You struggle to avoid them, but successfully survive the ride. You splash down into a pool of liquid. You've been washed into the stomach acid. Invasion challenge failed. Your detection by the immune system has led to your cellular destruction. Mission reset. Do you want to give colonization another try, or how are you going to access the body? Option A, by inhalation. I'm going to enter via the nose. Option B, by ingestion. I'm going in via the mouth. Option C, I spot a primary barrier weakness. I'm entering via a skin wound. Good spot. The skin is the primary barrier to infection. Any damage creates an opportunity for pathogen access. The blood supply is good, but there's lots of damage here. What do you do next? Option A, infection needs greater pathogenic load. I replicate. Option B, damage means immune response. I invade further to find an exploitable niche. So far, your replication is going unnoticed as the immune cells in the vicinity are occupied with clearing damaged body cells. A few of your progeny are cleared by phagocytes, but your colony is expanding rapidly. How do you want to proceed? Invasion challenge success. Success! You have successfully created a local colony of pathogens just like you. Soon your host will start to feel the effects. Mission reset. Do you want to give colonization another try, or are... Welcome back. I hope you enjoyed trying your hand at being the pathogen. What is the primary mode by which a macrophage engulfs pathogens? Option A, phagocyte. Right on. Phago, meaning to eat. Now that you've met some of the cells that mediate the innate immune response, I hope that you are ready for the next challenge, where you will negotiate the adaptive. Your next mission is trickier, and the cells you might encounter have some heavy-duty defensive tool. A final word of warning. The next body has met pathogens like you before. Its immune system has been primed with a vaccine. Be sure to make your choices wise. Tag the invasion portal when you're ready to take on the adaptive immune system. Before you start, you can choose one special ability for your pathogen that might help you evade the immune response. Which of these abilities would you like to take? Click the View Theory button to find out more about each. Option A, antioxidant secretion. Option B, hemolysin production. Option C, microbe-associated molecular pattern, MAMP shield. Good choice. Your microbe is now supercharged with genes for hemolysin production. This power-up lets you cut yourself out of membrane-bound vacuoles. Handy if you need to escape from a... Where do you want to begin your invasion? Option A. As you settle into the deeply gashed skin, you quickly pass through the epidermis and find yourself at the top of the dermis. The extensive skin cell damage has triggered... You negotiate broken tissue and cellular debris toward the deeper, nutrient-rich dermal tissue. The evidence of immune activity is all around in inflamed and digested tissue. A natural killer cell passes close by, but is not triggered by your presence as you do not express the surface. You manage to gain access to a capillary through a damaged wall. The blood is awash with antibody IgG that has been generated in response to the vaccine. There are circulating. As you scramble for the internal vessel surface, you make the realization that you do not possess useful cell surface components called adhesins that are necessary to adhere to surfaces. Invasion challenge failed. You are bumped along the blood vessel. Mission reset. Do you want to give colonization another try, or are you ready to return to base? Option A, return to base. Welcome back to base. I'm excited to hear what you got up to. Before we move on to the next challenge, I want to check how well your pathogenic invasions helped you appreciate the different immune cells. Which of these statements best describes the difference between adaptive and innate immune responses? You can write on. The B cells and T cells are triggered into action through specific stimulation of their receptors. The innate response does not require such specific activation. Which of these is not an important role that antibodies? That was a tricky one, but we must remember that it's the phagocytic cells that perform phagocytosis. The antibody is just a label that something should be eaten. Antibodies have so many important jobs. Their ability to bind antigens so tightly and specifically is one of the main reasons they have been harnessed by scientists looking. So in summary, the innate system cells work like first responders, performing non-specific clearance activities and raising the alarm to more specialist cells through cytokine signaling. The cells of the adaptive immune response regulate humoral immunity. I'm sure you encountered antibodies during your invasion challenge. These molecules are produced by B cells and plasma cells and are critical to many immune cascades. There are many types of antibody. As you may have encountered, IgG and IgM are two common types of antibodies produced in response to non-self antigens. This selectivity of antibody binding is something that has been exploited diagnostically for many years now. In our secret experiment mission, we'll see just how useful a tool antibodies can be. Congratulations on completing these two missions. You aced it. Swipe your card to move to your next challenge. Place your access card on the table. Welcome to the systems room. We are going to explore the two main categories of immune cells, the lymphoid and myeloid lineages. Are you ready to begin? I'm going to ask you a few questions about the cells of the immune system that you've met so far. I'll also introduce some new ones. You can respond to the questions by clicking on the... Okay, let's start with a question. In which of the highlighted organs do you think lymphocyte cells originate? Not quite. Remember, not quite. Yes, that's right. All the lymphoid cells start out as stem cells in the bone marrow. Click the common lymphoid project. B cells originate from precursor cells in the bone marrow. B cells express antibodies on their surface that can bind to non-self antigens to trigger adaptive immune responses to extracellular pathogens. B cells are important as without them, we would not be able to produce the antibodies which form the backbone of our immune system. You can find out more about B cells by clicking the View Theory button. T cells also originate from bone marrow precursor cells. T cells have specialized clonic receptors on their surface that natural killer NK cells are similar to T cells in function. They have different receptors to T cells, which allow them to kill cells that stop. Okay, let's check how well you're following so far. What cells are classed as lymphocytes? Click the View Theory button to get some guidance with the question. Option A, Cells that arise from a myeloid progenitor cell for macrophages and mast cell. Right on. Lymphocytes come from lymphoid progenitors, and myelocytes come from myeloid progenitors. In which of the highlighted organs do B cells mature? This isn't the correct answer. This isn't the correct answer. B cells develop in the bone marrow and go through 2 rounds of testing, one in the marrow and the other in the peripheral tissues. During central tolerance testing, B cell DNA undergoes a series of rearrangements that creates diversity surface antibody binding specificity in each unique cell. Which of the highlighted organs do T cells migrate to for maturation? This isn't the correct. That's right. T cells mature in the thymus. Remember, T cells are the ones that target infected body cells. In the beginning, T cells have no receptors, double negative, but then begin expressing 2 different types as they mature, double positive. What is the major functional difference between B cells and T cells? Click the View Theory button to get some guidance with the question. Option A. T cells are innate response cells, while B cells are adaptive. Option B. T cells attack invaders outside of the cell, B cells attack infected cells. Option C. B cells release antibodies to attack invaders outside of the cell, T cells attack infected cells. Option D. Trick question, they do exactly the same job. Great differentiation. This difference in approach and response is similar to the fundamental difference between humoral and cell-mediated immunity. More on this shortly. In which of the highlighted organs does peripheral tolerance testing take place? Not quite. Right on. Tolerance testing is a vital part of immune cell development where the B cells— check out the tolerance poster to your left. Tolerance testing is super important to stop your immune system from mounting a response to itself. In central tolerance testing, B and T cells that bind tightly to self-antigens undergo— peripheral tolerance testing is a second round of testing that occurs in the peripheral lymphatic organs, including the lymph nodes. Which of the following is not a likely outcome of a strongly self-reactive B cell being released into circulation? Click the View Image button to display the— that's right. Your immune system must be trained not to mount an immune response to normal healthy tissues and cells in your own body. Which cell can differentiate into a plasma cell? You're getting really fast at this. Plasma cells are what you get when a B cell is stimulated by a helper T cell to become an antibody-generating machine. Okay, that's the major lymphoid cell types covered. Now let's look at the myeloid. From which of the highlighted organs do myeloid lineage cells originate? That one was too easy. You know where the stem cells live now for sure. Myeloid lineage cells come from neutrophils are the most common immune cell, accounting for around 65% of all immune cells. They are easy to spot when stained in a blood test due to their odd-shaped nucleus that looks like— eosinophils are beautiful cells. They contain many small granules of enzymes and other chemical mediators that are released during inflammation. Generally, these cells are involved in clearing parasitic infections. You can spot them in stained blood tests due to their— basophils are very easy to spot. Their large, heavy granules of histamine and heparin are so numerous they make it hard to see the nucleus. It looks like someone filled a cell with grapes. These cells are important in recruiting other immune cells to the site of an infection or injury and can be involved in allergic responses too. Unlike B and T cells, these myelocytes tend to mature and differentiate in the blood. Did you meet any macrophages when you were on your infection and invasion challenges? Option A. Yes, they were the worst. I got too bad. Professional phagocytes are probably the most commonly active component of innate immune system. All invading pathogens will meet them sooner or later. I'm sure you will recognize this one. This is what a monocyte can become, a macrophage. These cells are specialized phagocytes, capable of consuming nearly any pathogen that it encounters, unless it's too big, in which case it might need some help. These cells are also... Can you remember this one from the invasion and infection challenges? It's a dendritic cell. This one is also capable of phagocytosis, but it doesn't eat to clear up. It eats to digest and then present parts of pathogens on the outside of its membrane-like metals. Well, there are many more cells in the immune system that we haven't been able to meet yet, but these are the main players to be... I'm satisfied that you have a solid immunological foundation and are the perfect candidate to help me with my global health mission. Swipe your card to move to the final secret task. Place your card on the countertop. Pick up the safety goggles. Find a glove box and click on it to wear gloves. Do you remember the antibodies IgG and IgM you encountered? We can measure the presence of different antibodies in blood sera via serological testing. If a person has previously received a vaccine to a particular disease and currently... Excellent. The only reason we might not see this pattern is if a person was unable to produce... The serum samples we're using today are sensitive to temperature. Being proteins, samples, and antibodies can begin to degrade if they get warm. Pick up the pipette to get started. You need to have a tip on your pipette in order to collect liquid. We automatically pick the tip for you. Okay, you're ready to get going. To detect IgG and IgM in the blood sera, we're going to use diagnostic antibodies that have been generated to bind with the blood Add anti-IgM primary antibody to the microplate. Now add anti-IgG primary antibody to the microplate.
To save us some time, I'll fill out the rest of the microplate's wells. We can collaborate like this every time. I'll finish the repeats after you've made the initial addition. Right now, I'm filling half of the plate with anti-IgM antibody and the other half with anti-IgG.
Discard the tip, hang up your pipette, and place the microplate inside the dark box. Placing the plate in the dark box during incubation protects the reagents from light damage, which can affect the validity of our experiment. The incubation is finished. Now we wash off the excess Open the dark box. Place the plate on the washer's tray. Great, the wash is done. We now need to remove excess. Grab the plate and tap it on the paper towel. Our plate should now be covered with antibodies. Our control samples are very important, as they place the plate back in front of you and grab the pipette. Add the required test samples to the plate. You can't pick anything up from this container. Great, it looks like you got all of them. Let me do my part now and fill the wells across the plate. Now that we have all the samples in place, we'll need to incubate and wash just like we did for the primary. This gives the epitope on the serum IgM and IgG time to bind to the respective capture antibodies on the base of the plate. I'll finish duplicating your additions if you can open the dark box. I'm all done. Place the microplate in the dark box. Open the dark box. Wash the excess sample from the plate. Looking clean. Now we just need to remove any remaining wash buffer from the plate before continuing with the last two antibody additions. Tap the excess liquid from the plate. Add anti-IgM secondary antibody to the microplate. Now add anti-IgG secondary antibody to the microplate. Incubate the plate for 1 hour in the dark box. You can't pick anything up from this container. Open the dark box. Wash the excess secondary antibody from the plate. By now, any IgG or IgM in the same place the microplate back in front of you, then pick up the TMB substrate bottle and half fill the pipetting reservoir with liquid. Now use the multi-channel pipette to transfer substrate to the plate and incubate in the dark box when you are finished. You need to have a tip on your pipette in order. You can't pick anything up from this container. Right. Half fill the new reservoir with sulfuric acid ready for pipetting. Open the dark box. Retrieve your plate from the dark box and use the multi-channel pipette to transfer sulfuric acid to it. Yay! That was the last. Grab the microplate to head to the analysis room. I'll get the door for you. The program is running now. The data will be sent automatically to the analysis room screen when it's ready. The data from the spectrophotometer has been delivered to the main screen. Go and check out the data on the big screen. There are a few extra patients here than we tested together, as I had already begun to process them before you arrived. I was so glad to have your help. Where we see a plus sign in the table, this means a positive detection has been made. Likewise, a minus sign means no antibody was detected. Ambiguous data is marked with a question mark. All of these patients come from a single community. We want to know how frequently this disease is occurring in the community's members. How many of these patients have either been exposed to the disease previously or have received a vaccine? Click the View Image button to display the wall screen image in the lab pad. Option A, 3. Option B, 1. Option C, 8. Option D, 7. Good interpretation. We're looking for samples with detectable IgG levels. It looks like Are any of the patients potentially suffering from a current infection? Click the View Image button to display the wall screen image in the lab pad. Option A. Yeah. The fact that we can detect disease-reactive IgM in these samples is highly suggestive of infection there. Should any of these samples be retested? Click the View Image button to display the wall screen image in the lab pad. Option A. Yes, samples 8 and 5. Option B. Yes, samples 3, 6. I think so too. Sample 8 might be a very recent infection. It would be a good idea to retest this patient in a few days to... Awesome effort with those interpretations there. This relatively small dataset is just... I've called up a bigger historical dataset that is identical in process to what we've just done here today. The difference is that this was the combined effort of thousands of researchers like you and me working in unison to keep the Earth healthy. You can investigate the data shown before you by clicking the tabs at the top, just like the display from the beginning of our challenges. The difference is that now you can also toggle another data set on and off. The base map in shades of blue represents the same data as before. Darker areas correspond to greater vaccination coverage. The slider allows you to toggle the number of disease cases in a given country on and off. The earliest date we've seen today is 1980. It's wild to think that since the licensing of a vaccine for this disease in the 1960s, the number of cases per year in the U.S. alone dropped from hundreds of thousands to being declared. Take your time to have a look over this data set. I've submitted the data from our experiment to support the next version of these maps. Raise the lab pad when you are done exploring. Have you checked out all the data? Option A. When many people within a close community are unvaccinated, it can create a situation where a disease can spread very quickly from person to person. This is how epidemics happen. The global health picture is nothing without the healthcare professionals and scientific researchers that collaborate to help keep all of our community. Let me ask you one last question.